Hello, everyone. Thank you for joining our webinar, The Fundamentals of Single-Cell Sequencing. For those who just joined the webinar, a very warm welcome. And our speaker today is Dr. Sujata, Singleron's Regional Application Manager. She'll be sharing on Singleron's single-cell sequencing technologies from sample processing to data analysis. If you have any questions, you can use the Q&A function at the bottom of your Zoom window, and we will address all Q&A questions after Dr. Sujata's presentation. And hence, without further ado, let's invite Dr. Sujata.
Once again, thank you everyone for joining this webinar session. Appreciate you taking the time. So today, the main agenda is to kind of understand the fundamentals of single-cell sequencing. But before we kind of dive into that, I thought we could just set up a bit of background understanding on what single-cell sequencing helps with and how things were done traditionally. So if you think of like a biological sample, which can be similar to a fruit bowl, traditionally, a smoothie-based approach was used. So if you make a smoothie of this fruit bowl and you sample it or you drink it, you can get a feeling on what are the different fruits that went into it, but you're probably going to taste the most dominant fruit in that. More advanced technologies like single cell actually helps you sort of interrogate that sample or here the fruit bowl by actually picking out the different fruits in it. So here you know what different fruits went in clearly. And then you can also count how many pieces of each fruit is there. So if you think about a very rare piece of fruit that was in the bowl, you can actually get that information by using single cell technology, which you probably missed out when you smoothied that fruit bowl. So moving from that example to actually a real biological sample. So if you look at, say, a tumor, which we know has various different cell types. So you have the sort of malignant cell type in the tumor. You'll have immune cells, stromal cells, blood vessels within the tumor, fat cells, neurons, and non-tumorous cells of the same cell type. So all of this form this tumor microenvironment. And each cell type has its own sort of gene expression or signal. So certain genes would be upregulated, certain genes downregulated. Using sort of a smoothie approach or bulk RNA-seq approach, you would sort of average out all of this signal. And so your understanding of how each gene is upregulated or downregulated can get a bit sort of missed out. So you will be looking at a sort of average profile here. But if you can actually find a way to separate each of these different cells and then look at the gene expression, it becomes pretty evident that you can see the expression of every single gene in every cell. And this gives you more information, not just on the different cell types in the sample, but an understanding on how these cells interact with each other and how they interplay in such an environment. And so you can look at single cell sequencing data as a high resolution catalog of all cells in sort of a tissue or living organism. And with that data, you can ask questions as simple as what are the different cell types in the sample. Going to more complicated questions is how is gene expression shifting at the single cell level between the different samples. You then move to comparing different kinds of samples. So example, you've got control, you've got a treated sample, a disease samples. So the way you think of it, you can actually ask more and more insightful questions by using this big data set. And so it's not surprising that single cell is sort of almost staple in a lot of different fields now. You do single cell data sets for atlas studies, which is just kind of having an in-depth understanding of a particular tissue. It's very relevant in immunological studies when you're trying to understand immune diseases, how is the immune response to these infections. In the field of developmental biology, understanding development processes, what are the different stages cells go through. Then in any kind of drug development, you need to know, identify biomarkers and then targets, and then investigate the effects of these drugs on different cell types. And finally, it's not just now restricted to human or say mouse model work. Single cell is applied to all different kinds of organisms, zebrafish, plants, etc. Before I go into the talk, I just want to point out two things just to keep in mind. So when we talk about single-cell RNA-seq, usually we are talking about profiling the transcriptome or the mRNA. So these is mRNA, the genes that get translated into proteins. At the single-cell level, there are many different layers of data that you can sample with different assays, and I will touch about a few of those later. But for the majority of it, when we say single-cell RNA-seq, I'm talking about the transcriptome being profiled. And another image that you will usually see in single-cell talks is a UMAP, or a uniform manifold approximation and projection map. And so the way to just understand this is that it's a very simplified pictorial representation of a complex single-cell data set that comes from a single-cell workflow experiment. So what you need to look at is every dot here is a cell. So it's data from a cell. Every color that you see in such a map means a cluster of cells. And so the closer the dots are, the underlying data is similar. So in this case, their transcriptomes are similar. And so they're closer and closely clustered. And then once you have these clusters, you can use highly expressed or marker genes to identify the cell type. So you will usually have names of different cell types on a map like this. Okay, so once you've decided that you want to do a single cell or a single nuclei project, the first thing you need is your sample. So if you're setting out to do a single cell project, you can use fresh tissue. So these can be harvested or biopsy samples. You can also work with organoids. So these are more artificially created samples. You can also work with cryopreserved cells. So if you can work with PBMCs or any other cells that are cryopreserved. And what I've shown here is a very standard single cell workflow. You start with preserving this tissue that you collected. You need to dissociate it into a very good, high quality single cell suspension. Then you need to find a way to actually partition that to individual cells so that you can actually capture the mRNA at the single cell level. You sequence all of this data, all of these cells together, and then you have data analysis to tease back to understand from which cell, which sort of mRNA has come from. Now, say you've actually flash frozen samples. So this is sometimes you've got like a clinical cohort and these samples have already gone into the -80 freezer. Not an issue. You may not be able to do single cell, but you can actually do single nuclei sequencing. So if you look at the workflow, the first part of it's slightly different. You just kind of go in and isolate the nuclei. But from here on, you can actually just follow a standard single cell workflow from partitioning all the way to data analysis. So for today's talk, we'll kind of go through these 4 steps, starting with to collect and preserve your sample. How do you prepare a good single cell suspension? What are sort of the sequencing workflow you can use? And then finally, what does data analysis for single cell look like? So for starting off your experiment, you need to preserve a tissue for either say you want to store it because you've got a lot of samples to collect, or you have to transport it from where it's actually been harvested to a lab setup. You also need this solution that you work with to preserve the viability of the cells. So for single cell sequencing, your cells have to be alive. And so for that, it needs to be in an environment that sort of mimics the physiology of the living sort of setup. And so we have a tissue preservation buffer solution. So the one that we have on our portfolio is called SL Live. And so this kind of helps you preserve your solution for about 3 days at sort of 2 to 8 fridge temperature. And so there's no fixatives in this. So your cells and a sample remain fresh. And so here's some data to show that the viability is maintained quite well over 3 days. And the way to make sure that your cells samples are not impacted by preserving is to check what are the different cell types across the whole sort of 3 days time. And so we have some data here to show that the different cell types are correlated, which means they are the same if you sample them after 1 day, 2 days, or 3 days. So once you've got your tissue collected and preserved, then you can move to the next step, which is to prepare a good single cell suspension. So here comes tissue dissociation. So this is a sort of a stage where you need to have an option where you can dissociate your tissue. And you want to be able to dissociate a broad range of tissue types. So it should be a sort of option that you're using that can work across different tissue types. And of course, different tissue amounts, because it's possible for every sample, you're not going to get a lot of tissue. And you also want to work with something that can do dissociation in a short operating time, because the longer you keep it at this stage, the faster the cells are going to die. So you don't want something that needs to run for a really long time. So at the end of the tissue dissociation step, the outcome you're looking for is a suspension with really high viability and a good yield. So a lot of cells from that sample. So you're getting an unbiased mix of cells. And so from tissue dissociation, you can also go into the single cell workflow, which I'll be talking about, but you can also use that single cell suspension to do any kind of flow studies or cell sorting or single cell culture. And so here I would introduce our tissue dissociation option, which is the Python. So we have two. One is Python Junior, which is a small 2-channel automated option for you, or the Python I, which is an 8-channel option. So dissociation here can be carried out, it will be carried out with two different sort of setups. One is an enzymatic part, which is based using enzymes from our SL Live tissue dissociation kit. And the other part is a mechanical dissociation, which is done with the instrument. And so we have this little unit into which your sample and the enzyme goes in, and then you can actually use the dissociate to dissociate your sample. The enzymatic dissociation reagent, of course, you need it to be validated across various different tissue types, which we have. And this is because you can then use this universal solution across many sample types. As you can see here, there's more than 300 different tissue types that it's been tested on. And the data here, when compared, you can see that the cell viability, which is an important factor at the end of dissociation, is really high for the sample if run with this particular kit. And an instrument actually helps you keep consistency across replicates. So if you're doing a lot of samples for your project, then it's a good idea to have an automated option so that every time you're working with the sample, you get the same consistent cell representation and data. Another important thing is to make sure that your dissociation option can actually work with really low amounts as well as large amounts of sample, because that depends on what sample you get that day. So you can see here that for our system, we've actually tested as low as 10 mg, which means even small biopsy samples can be dissociated in the Python. And so we have a very fully customizable and plug-and-play set of protocols. The temperature of this instrument is set at 37 degrees for tissue dissociation. It can run between 30 to 300 RPM. And just as you see in the video, it loops counterclockwise and clockwise to actually mechanically dissociate your sample. So the programs kind of have a few parameters that can be tweaked, and they are fully customizable depending on your tissue type. So if it's a very hard tissue type, you can actually increase the speed and the time. If it's a very sensitive tissue type, you can try to keep the speed low and do a quick dissociation. So this is a very flexible option to use. And just using the instrument is also very straightforward. So you can see the workflow here. You open the unit, add your solution and tissue based on the amount and the weight. You assemble the unit, put it into the instrument, and run your program. And then at the end of it, you get the single cell suspension that can be taken out for your downstream processing. Of course, depending on your application, you might have to do a few steps before you actually get started with your single cell cell partitioning workflow. So it could involve filtration based on size to remove some clumps. If you've got a lot of RBC, you may have to remove that. And if it's got a lot of debris at the end of dissociation, you also want to think of a way to actually remove a bit of debris. And then you run a QC, you make sure that you have a good count, a good number of cells in the solution, and then they are highly viable. And so here's some data. This is showing how different tissue types have been tested on the Python, and you can see that the viability is very high. Of course, the cell numbers will vary depending on the input material used. And then you can also see that some tissue types are smaller, giving you less number of cells, where others will give you more. And all of these samples can be run on short operating time between 15 to 30 minutes on the Python. So before I move on to the single cell part, the question it would be, do you really need an automated dissociator, or can you actually just use manual ways to dissociate your sample? So here's just some data showing that if the same sample is put through a manual dissociation and a Python Junior or automated dissociator, you can see that though viability from both are quite high, you get a lot more cells from the Python Junior dissociation. And what that means is you're actually sampling better, the tissue is sampled better, and you see more tissue cell types that you would miss if you only did a manual dissociation. So all in all, it's a good idea to think of an automated way of dissociating a sample for improved cell yield, more cell types and subcell types, and then you get no compromise in the gene expression profile. So I just want to showcase some publications that have used both the enzymatic and the mechanical dissociation Python. And so this is one is in mouse tissue sample and the other one is in human samples. So now just moving down to the next step, we're at cell partitioning and capturing the mRNA and then preparing a library for sequencing. So at this point, what you want to look for is a method that can capture all different cells from all different kinds of samples and all cell types without any bias. You also want to capture the rare cell types because those are also important, and also capture sensitive cell populations. So some cell populations can die faster or they get stressed faster and may not survive the single cell workflow. So you want a method that can actually keep these populations when you're running a single cell workflow. And so that brings us to our technology, which is scope chip. This is what we use for partitioning the sample and then capturing the mRNA. So as you can see, it's sort of a slide-like setup, and it's a very gentle setup because we just kind of pipette cells into this chip and let gravity do the work with the cells just falling into wells that are on this chip. So we've got a few kinds of chips. So the standard chip has about 150,000 wells, and we can capture up to 20,000 cells there. And then we also have other iterations that can capture larger or more number of cells. The other part of our technology are barcoding beads. So these are beads that actually have oligos that have unique cell barcodes and a poly-T. So as I mentioned, our capture here is mRNA, so which all have poly-A. So we use a poly-T oligo to capture the mRNA. So all you need to do is add your cells into this chip and then add your beads. And once the cells are lysed, all the mRNA from the individual cells get caught in these beads, which have unique barcodes, which can then be used in data analysis to allot them back to the cell that they came from. As you can see, we also have streamlined this in an automation option. So you can either do this whole workflow on the scope chip using a pipette by loading it one by one, or you can use an automated option that's known as the NEO, where all of these steps can be automated for you. And so the two sort of kits that we have for this are single cell and single nuclei. You can use this for all different cell types or tissues, choose to do manual or automation, as I mentioned, and capture up to 30,000 cells. Just to kind of showcase that these kits, single cell profiling, RNA profiling kits have been used in various different sample types in humans. So these are publications on lung tissue, colon tissue, PBMCs or breast cancer tissues, but we've also kind of, you can see that these kits have been used in mouse models, in Drosophila, etc. So it can be used, it's species agnostic and can be used across all different samples. And so what are you looking for when you're doing single cell work? How do you know that your sort of setup is going very well? Is like you're capturing rare cell types in your sample. So here, if you look at a UMAP, like I mentioned, you can see that you pick up even immune cells in an embryo, which are a very small population that you can see here. So this means that the capturing efficiency of this kit is really high. And then you also capture sensitive cell types. So neutrophils are known to be quite sensitive to any sort of manipulation. And you can see that because our chip and microvels loading technology is very, it's not very harsh, you actually capture sensitive cell types. like neutrophils. And so just to kind of showcase a publication here where you can see that neutrophils have been captured from these pancreatic tissue. So just to kind of come back here, I also want to just quickly mention about the single nuclei workflow. As mentioned, if your samples are all flash frozen, you can do the same thing. Once you isolate your nuclei, you can use these kits to do the remaining part of the workflow. And then sort of just to kind of step back and just kind of go through all these products that I spoke about. Here's a kind of study where they've used a tissue preservation buffer to preserve samples from a mouse. Then they've used dissociation to get a single cell suspension from the liver, did some enrichment of immune cells, and then did a single cell sequencing. And so using this, they were able to actually understand the mechanism of some immune escape from certain cells. And that kind of gives them information to design new therapeutic strategies. Similarly here, if an example of using organoids, so this is an organoid starting material. Again, they're trying to understand a mitochondrial disease. You kind of dissociate them, do a single cell workflow, get your UMAP to understand all of the different cells. And from there, they've used more advanced analysis like pathway analysis to understand the disease and what mutations could be causing them. So we also spoke about nuclei kits. I want to kind of mention sort of some samples in which doing single nuclei might be better. And these are usually adipose tissue or brain or muscle tissue. And the reason for this is that some cell types in these tissues are quite large and may not be captured if you did a single cell technology. When you do single nuclei do not have these biases on sample cell sizes. And so you capture some of these cell types that you would miss if you did single cell. So if you look at these two UMAPs here side to side, you can see it's the same sample that went through single cell and single nuclei workflow. And you can see that there's certain cell types that I've circled here. That's the myocytes, which is actually the skeletal muscle cells. They are missing in the single cell data. And that's because they are kind of irregular, large shaped and may not get captured. Whereas if you did a single nuclei workflow, they get captured. And so depending on the sample you're working with and the question you're answering, you can decide which one is better for your setup. And so here's sort of a publication that was interested in looking at muscle tissue. So they actually did single nuclei, and you can see that it gives you the same kind of data. You can do your UMAP and find out the different cell types in your sample, and then do other analysis, just as you would do it for a single cell setup. So what we talked about now was mRNA profiling. I just want to also bring up that today, for now, we actually have many more options, many assays which profile not just the mRNA. You can put an additional layer of data. And these are just some of the options available from our portfolio. So as you can see, you can profile the mRNA plus the immune receptor. So these are kits that are sort of relevant in immunological studies. We also have option to profile other sort of non-poly A mutations. So that's when we talk about, because for the transcriptome, usually we are talking about poly A mRNA. But say you're interested in something that doesn't have a poly A tail, then you can actually use a kit like Focuscope to capture that kind of information. Or if you're looking at something more like glycosylation levels at the single cell, you can also use, there are assays available for that. So I'll just go through this in sort of just a highlight, and then we can always later discuss or have another session to look at all of these kits in detail. So if you're actually interested in looking at the immune receptor profiling in samples, you can use kits such as S-Circle, where you're actually able to get the whole TCR or BCR. So these are the immunoreceptors of T cells or B cells, and you can use that information along with the RNA information to understand clonotypes in samples. So for example, this particular publication, so they're looking at human hypopharyngeal squamous cell carcinoma. Not only do they get all of the single cell information between the different samples that they work with, they also want to get an idea on what is the TCR profile of each sample. So this is what are the T cells that are there in each sample, and then they can kind of understand what was the immune reaction or what they can work with for any sort of treatment, etc. So this is sort of an additional layer at the single cell level that you can add to your study. Another one is focus scope. So here, if you've got any interest in single nucleotide alterations or mutation or gene fusions, these are sort of, again, another layer of data you can add with the mRNA profiling. Also, as I mentioned, if you're interested in any non-poly A transcripts, so this is if you're looking at viral infection in single cells or intracellular bacteria, we have options to look at that as well. So how would that look like? So here's a study that used a blood cancer panel. So this panel, along with the mRNA at the single cell level, can also pick up these different mutations and gene fusions that are important for in the particular disease. And so if you look at the data, you've got like a UMAP showing you all the different cell types, but you can actually go in and find out which cell types have these mutations. And then from there, you can actually think of how, what is the kind of therapy that you can develop based on this. And another kit I just want to kind of highlight is a Mobioscope. So here we're actually looking at a kit that can give you more coverage of every transcript. So this is if you're looking for more genes, more splice isoforms, interested in understanding alternative splicing sites, or you're working with non-model organisms that don't have a very well sort of annotated genome. So for all these cases, we have also an option where you get more than just the data from that you would get from a single cell kit. So this is also something that's available to think about. So once you've come down to the workflow and you have your libraries ready, from there you actually you send it for sequencing. All of our libraries are compatible with most short and long read sequencing platforms. And then once you get the data back is when you move into the last part, which is data analysis. So we have a few options available to analyze your data. I will go through the first one, which is Telescope. So this is pretty much data pre-processing. So what you would get from a sequencing provider once you sequence your libraries is a lot of sequence data. And in some way, you need to use a reference genome for that particular sample. So species that you're working with. This genome with gene annotations will help you get a count matrix, which is the basic sort of data that you will work with for any downstream single cell application. So this is possible using Telescope, which is a free to download software available. And once you get to this stage with your count matrix, you can then think about what are the questions that you set out to answer. So in this stage, you want to analyze data, differential gene expression, pathway, cell-cell interactions, etc. These are standard single-cell analysis modules that you would do. You would also want to visualize the data, which is where your UMAP would come in. And then if you're working with multiple samples, you also want to integrate the data. So the figures here are sort of standard analysis that you would do for single-cell work. Now, you could code all of this in RStudio if that's something that you've got expertise in and you can figure it out. But if that's not the case, we also have a automated sort of cloud-based data analysis platform available. So this makes things way more simpler. You can actually upload your data. So there's different formats depending on which sort of data set you're working with. You can just upload the data and then do any kind of QC all at clicks of buttons. And then once you set up the analysis, any kind of sort of filtering methods, etc., you're able to get all of this in just a click of a few buttons. You get a cell annotation. So as you can see here, the sample is annotated for you based on underlying database. You can look at the cell composition across your samples. And if you're interested in looking at particular genes, you have that sort of option here, depending on the gene of interest. What are the cell types and clusters that have high expression? What are the genes that have low expression? And you can also do like heat maps to kind of understand differential gene expression. So this sort of cloud-based analysis software helps you to generate all these images, and it also helps you download and use them for any other sort of papers or posters, etc. So it's quite easy to use. And so just to kind of step back and look at the workflow, as mentioned, we start from tissue preservation for a single cell workflow, do a dissociation, do a partitioning and mRNA capture, you sequence your libraries, and then finally use bioinformatics to put all of your data together. So we've kind of covered all of this today. we also have a end-to-end single cell analysis service lab in Singapore here, where we're based. So you can send samples here using you can use our preservation buffer, and then we can actually run through this whole thing and then provide you with your final results with bioinformatics support. Or you can also connect directly with our trusted partners. Depending on where we are, we have collaborators and distributors who are ready to support you with all of these products, depending on the need and the question that you're looking to answer. And so before I end, I'm just if all of this has been new to you and you're still planning your experiment, or if you've done some bulk and you're not quite sure where to go, just to think about how all of this falls into a project plan. So I spoke about bulk RNA-seq and single-cell RNA-seq. So you can kind of think of using bulk RNA-seq, or you've already used it for some kind of initial screening, like you did some bulk comparison between groups. You can actually use that information, and if you've discovered something, certain genes, etc., you can then use single-cell sequencing to further dig into that and understand like the cell types that could be playing a role in that genes that you discovered, or subpopulations, and then try to look at gene expression, the phenotypes related to that. So this is kind of an option where you already have bulk data, and from there you can actually zoom into doing single cell work and get further insights into your data set. The other option would be to actually do a single cell first. So you kind of want to start with single cell, where you look into something and try to identify something from a small subpopulation. So say, for example, you've characterized a biomarker using single-cell RNA-seq. And from there, you can upscale and do more bulk work to validate that, and then use predictive models available today to see if this works. So these are the two ways you can think about planning your project. If you've not started, or if you've already done bulk, you can think about how single cell can actually come into that project and give you better insights into your data. That's what I have for today. I hope you got some idea on how single cell, the fundamentals of single cell, and if you were thinking about doing it, a little bit more information on getting started. And I'm open to any questions.
Thank you very much, Dr. Sujata, for the very informative presentations. As mentioned just now, you can post your questions by clicking on the Q&A icon at the bottom of the Zoom window. With that to say, we actually do have a few questions. Let's start with the first one. Someone asks, if we do this in a smaller cohort and still wants to validate on a larger cohort, which will be the best technique to use? Is single cell suitable? Things like that.
I would think single cell is suitable, but I probably need a little more information on what's the question and what you're looking at. So I think I will. Is there any more info on that?
So far there's no. So
Okay.
for
So,
for Miss Sujata, it's another Sujata online. So
okay.
yeah. So for the person who
Okay.
asked the question, maybe we can email you, or
Yeah,
if
maybe
you
you can get in touch with us, and then we can help advise on what's the best way forward.
yeah. Yeah, so because that's the only sentence that she typed, so I think we do need a little bit more context.
Sure.
Yeah. So there's a second question that asks, is there any data showing that epitopes on the surface of single cells are well preserved when we are using Python?
Okay, so there is depending on the tissue that you used and the marker, it depends on the solution that you use for dissociation as well. So we have a universal solution that usually should give you a single cell suspension. For the markers, we also have a more specialized product known as the dissociation for flow, because that's where surface markers are important. So we would usually recommend trying both and seeing what works better, because they kind of they sort of have two different purposes. One, require your tissue to break down into single cells. So the enzyme might be harsh for a surface marker. So we have another kit that helps you preserve these epitopes. So in general, my recommendation would be to try both and then see which one works best for your sample type.
Okay, thank you, Sujata. Someone else also asked, what is the minimal amount of samples that we can start for single cell sequencing, and also how to avoid noise disturbance during analysis?
Okay, so I think minimum number of samples, it kind of also depends on your experimental setup. But say if you're doing, say, control versus a sort of disease or treatment model, usually you want more samples to improve your sort of statistical power. So this comes when you're doing analysis. The more data points you have, the better to understand and do statistical comparison. So I'd say at least 3 is a good start. Of course, that also depends on sample availability and how much sort of funds you have to do this research. So this is a kind of guideline to do at least 3 if you're doing. But even if you have less, I think you can gain insights by running a single cell experiment. Sorry, Irene, what was the second part of the question?
Second part of the question is how to avoid noise disturbance during analysis.
So noise disturbance. So in terms of analysis, so there are a few sort of uh, areas from which noise can come in your data. One could be you've got so a doublets or multiplets, or you've got some kind of ambient signal coming in. For most of these sort of known sort of noises in single cell data, there are algorithms available, published sort of scripts and suites of analysis that you can run your data through to make sure that you are discounting or removing most of the noise and keeping only real signal. Of course, it helps when you have more data points again, rather than less, to help understand what's real biological signal and what's background noise.
Okay, thank you. There's a lot of questions. We are running a bit short of time, so I'm just gonna pick another few, and then for the rest of the audience that we did not manage to answer here, we will definitely come back to you via an email instead after this webinar. So there's someone who asked as well, like on plant plasma cells. So this just now, as mentioned, some of our kits are actually feasible for plant cells, and they are a bit concerned because plant cells has very high chlorophyll and high biochemical content. So they just want to know how how Singleron's kit goes well together with our plant cells in general.
Right. So the optimizing for that part actually would kind of be similar to tissue dissociation and optimization. So for plants, usually single cell workflow is done either on protoplast or nuclei. So you kind of have to make a decision which one you'd want to work with. And this is also probably dependent on how easy it is to isolate either of these in the species of interest. So once that part is optimized, you can actually just load either of this into our chip, and then the remaining single cell workflow is quite straightforward. So I'd say it's more about figuring out how to optimize that first part. We can support with that on seeing what's the best way to get a good quality protoplast or nuclei suspension.
Mm. Okay. Someone also asked, how are our technology different than the others in the market?
Okay,
Yeah.
so I think I can kind of maybe just if the most popular technology is a droplet-based technology, in which you need an instrument, and the technology actually uses sort of a oil emulsion to make a droplet with the cell and a bead. So that's a droplet technology. Ours is a microwell technology. So uh, can you see me? So we have a chip. So
Yeah,
that's
we
what
can.
our chip looks like. So it's wells here, and you can actually just add the cells. So it's quite different from the other technologies in the market. And as I mentioned, there are some advantages to using a microwell-based stress-free technology in the data that you capture at the end.
Thank you, Dr. Sujata. Okay, one last question. So someone asked that for sickle cell sequencing, because our beads are attached to the tag and used to label them. So how does this work for single nuclei sequencing technology for frozen samples? Since also because the isolated nuclei will stay separated, or will they aggregate during the sample prep? I think this is something that the audience would like to know.
Okay, so if once you have a single cell or nuclei suspension, they can both be sort of clumping can happen in either. So that depends on actually making a good suspension to make sure it's clean, filtered well. And once you have that, loading it is the same. We have a different buffer to lyse the nuclei when compared to the cell. So that will be a slightly different reagent. But post that, you are using the beads to capture poly A. And so it's the same sort of transcripts that will get captured that have a poly A. I sort of showed in one of the slides, because it's the nuclear, you will capture about lesser number of mRNA, because the amount of mRNA is probably about 30 to 40% of what it would be in a single cell, but the capture method is exactly the same. So what you have in the nuclear, you will capture by the same method, but it will be slightly lesser than single cell just because the nuclei has less mRNA comparatively.
Okay. I think that concludes the end of the Q&A session. Once again, thank you very much, Dr. Sujata, for, having us, and same goes to the audience in our Zoom call. Very good. Very happy for you guys to be having like 1 hour afternoon with us. So with that to say, we are getting to the end of the webinar. If you have any more questions on single cell sequencing, do contact us on sales@singleron.bio. You can see the email, which is right above the LinkedIn QR code. Yeah, you can also email by copying us from the chat as well. Follow us on LinkedIn and X to stay tuned, and to also help us improve. Please take 1 minute to fill our feedback form at the end of the webinar. And with that to say, thank you everyone for joining us today, and we'd like to see you in our next event. Thank you.
Thank you.